Now, after geometry, let's switch to other science. I'm very glad to introduce my colleague, Manuela Marai, who will present on the science in Galen's pharmaceutical science, the case of wound treatment. Manuela, please, I'm leaving the floor to you. Here's the microphone.
Thank you. Okay, thank you very much. Thank you all. Thanks, Simone, for putting this together, and thanks for having me here and for your hospitality. I'm very happy and grateful. So what I'm going to talk about today is basically what I've done in this past 4 years. So what I did and how I did it and why. So basically I wanted, and this is a clear example of how you need to, if you're actually interested in the science, when it comes to ancient science, you really have to break the boundaries. It sounds obvious, but it's not really obvious to be interested in the science of ancient science. So I wanted to study Galen's pharmacology, and when I met my future supervisor, Caroline Petit, and then she said to me, well, you are a scientist also, you're a biologist. So you might want to work with this person at Warwick, who's Freya Harrison, who's a biologist, and she has been she's been searching for antimicrobial substances in medieval medical texts. And she put together basically a team of linguists and historians and scientists to investigate, to search for, it's a practical application of this case, medieval texts. And so the idea was, she's been working on this medieval recipe, which turned out to be a very, very powerful antimicrobial once you mixed all the substances together. So that's the beauty of it. It's the combination that produces the antimicrobial activity. And so the idea was, okay, we can do the same in Galen. You search for antimicrobial substances in Galen's medical text. And this is somehow, it's part of a trend. It's kind of an historical approach to ethnopharmacology. Ethnopharmacology is the idea of finding scientific evidence for drugs that have been used in traditional medicine since forever, but also the idea of finding something new that is not used anymore, and it's there, it's the ancient text. And so there's been a reassessment of ancient materia medica, because it turns out that many substances used in traditional medicine and ancient medicine are actually effective. And also, the ethnopharmacological studies are mostly come from traditional Chinese medicine and Indian medicine, but there's been a reevaluation also of Greco-Roman medicine. So, for example, we have the antibacterial properties of clays and combination of substances used to treat pain, and up to a very recent book written by Nick Summerton, who's actually a retired physician, who wrote a book on how Greco-Roman medicine can teach us today. It's not just about pharmacology, it's a general approach to the patient. And some of these studies actually insist on the fact that it's the combination of the ingredients that makes the remedy effective. I'm sorry, because this is, I think it's the PDF, so it's slightly different than the PowerPoint. When you talk to classicists, you talk to classicists and you say, okay, this is what I want to study. And then they look at you like you're crazy. And because they start, obviously object, and they start, asking questions and posing problems which are there. You cannot really deny them. So they start talking about, yes, but what about the substance identification, because the species, the name and the species, even the species nowadays is the same, and then the chemistry of the plant can be different. And then what about the retrospective diagnosis? Because is the disease the same that we have in the text and that we have today? The symptoms might be the same, but we might have the same disease, but the symptoms are different. And then you have, yes, but what about the efficacy? Because in antiquity, they might have different expectation. Maybe they didn't really expect to be cured. They just want to get better slightly. And then you have the placebo effect. And then you have, for example, this idea that there is also part of a social dimension, that efficacy is something which is perceived, but it's not really real sometimes. So, but the thing is that even if all these must be included in the analysis, the fact that there has been a reassessment of the efficacy of ancient pharmacology starting from the 80s and the 90s with the studies by John Riddle and his commentary on Dioscorides, so the pharmacopoeia, ancient pharmacopoeia, and Cameron studying Anglo-Saxon medicine. These authors, they have a medical background. And so they started saying, well, this is not just a bunch of nonsense. You know, there's actually some rational use of effective substances. And also there's been experimental assessment. So the fact that actually they went to the lab and actually tested some substances and they said, okay, these are effective. But there is also this idea, which I insist on, the fact that it's important to acquire an historical perspective, because we do have, significant drugs nowadays that actually has a long history of use. Sometimes they were used in antiquity and then they disappear and then they came back, but sometimes the use has been there, for centuries, if not millennia. And also, surprisingly to me, when it comes to natural substances, a very significant percentage of drugs that have been marketed in the past 40 years, they have somehow a natural origin, which means that they are not really, it's not like a plant extract. So it's an active principle which has been modified. So it can be semi-synthetic. It can be something which mimics somehow a natural molecule, but it's a very significant percentage. So it's not all synthetic, basically. And Riddle, in his commentary on Dioscorides, when he talks about the methodology, he says something which it sounds obvious, but again, it's not. So this idea that we cannot, when it comes to ancient pharmacology and medicine, we cannot be disinterested in the efficacy, because somehow we need to understand Dioscorides' contribution to the history of medicine. And then on the other hand, you cannot really avoid the things you know today when you read an ancient text. You cannot really avoid the modern notions that you have in science, which are actually sometimes very helpful when you read the text. But also when it comes to the efficacy, it talks about the efficacy. So it becomes really boring if you just look at the substances that were used in antiquity and you say, okay, they are still used. Okay, that's it doesn't make an improvement to the understanding of ancient science. He's specifically talking about efficacy of the substances, but I wanted to take this further because he talks about Dioscorides, who wrote a pharmacopoeia. So it's a list of substances and what they were used for, but there's no theory. While in Galen, there's a lot of explanation and the rationalization of why substances were used and why they were combined, what's the function of the substance within the formulation, and how they affect the body, how they interact with the body. So Galen is a completely different animal when it comes to understanding actually the science. So my idea was, okay, let's look at Galen's pharmacology, so we have seen how Galen wrote a book on simples with this pharmacological theory, but there's also the pharmacopoeia. I was interested in formulations, because that's when the magic happens. It's when you combine the ingredients that you might have the effective drug. And I was focusing on antimicrobial substances for basically pharmacological and medical reasons. And I wanted to study wound treatment because I don't have the issue of a retrospective diagnosis. So wounds are wounds and they have a high infection risk. And Galen wrote a lot about wounds. And of the two works by Galen on compound remedies, one, which is compound remedies according to types, is specifically on wound healing plasters. And it's actually propedeutic to the other one, because in this work, Galen talks about the method of compounding. And nobody really in scholarship, not many people care about compound remedies, and nobody cares about this book in particular. They tend to focus on the second one. So there are previous studies about wound treatment in antiquity, but they have a lot of gaps for several reasons. They have some limitations. So when I started studying, when I started reading, started browsing through these compound remedies books, so which are basically recipe books, but that's a very limited definition, because there's a lot of theory in there. So you start reading and okay, there's a long list of ingredients, but then you have talks about the classification of the plaster, talks about like warming, cooling, drying, and moistening, and then you have cicatrizing, and then you have something which is enfleshing, which means it regenerates the flesh, and then you have different type of wounds, and nothing makes sense if you start from the pharmacology. In the same way that if you're a student of pharmacology, you don't start from pharmacology, you start from chemistry and physics, and then the physiology, and then the pathology. And Galen insists a lot everywhere in his corpus about the propedeutic reading. So what you need to read first in order to understand what comes after, which sounds obvious again, but it's not. So when I started reading the book on compound remedies, and then I started going back and actually following Galen's curriculum, Galen even wrote a book on the order of his own books to explain what you need to read first. So you start from Mixture, which is the work on the natural philosophy behind the physiology of the body. And then you move on to the books on pathology. They explain the classification of diseases according to the causes and the symptoms. And then you move to the therapeutic method, which is kind of a huge monumental book on the principle of treating disease. And then you move to pharmacology, starting from simple ingredients, and then you go to types, which is propedeutic to places. So that's what you need to do. And that's basically what I did in order to understand what Galen was talking about. So you need to put Galen's pharmacology, Galen's pharmaceutics, okay, so the formulation, into Galen's system of medicine. So you start from Mixture, and you learn that you have different mixture of elemental qualities in the body. Each bodily part has a different mixture of hot, cold, dry, and wet. And when you have an imbalance of that, that's one of the causes of the disease, but that's not the only one. You have anomalies in the bodily fluids, which are the humors. But if you call them humors, sometimes it's you don't really understand what it's talking about. They are bodily fluids. And then you move to the pathology, to the other books, and where you learn that the causes of the diseases are actually different depending if it's a tissue or an organ. That's a simplification, but. And Galen, when it comes to wounds, it's quite a novelty, but he classifies the wounds as a different disease, which can affect both tissue and organs, and that's a dissolution of the continuity in the surface of the body, and can actually be related to an anomaly in the fluids, in the bodily fluids as well. And then it really helps if you know what happens in what we know today about what happens in the wound, and how they are assessed today, okay? Because Galen insists a lot on the classification of the wounds. So today, wounds are assessed according to certain parameters, which are mainly the thickness, so the tissues involved, the complexity, if it's fresh or old, if it's chronic or acute, and the origin. So if it's from a trauma or if it's from an internal somehow an internal pathology, so to speak. So if it's like it's usually a vascular insufficiency, so you have varicose vein or you have diabetes, you can have sore beds. Okay, so those are different. The origin is different, and a wound must be treated differently. When it comes to what happens in wound healing, basically you have an inflammation phase where you have this fluid leakage from the blood vessel that helps somehow. You have proteins and white blood cells, so try to kind of start the inflammation process and fight a possible infection. And all these proteins, they attract other type of cells, which should then develop into muscle cells and tend to kind of close the margin of the wound together. They pull basically the skin. And then you have the formation of a connective tissue, which is then replaced by a scar. So this is basically what happens. And when it comes to favor the wound healing process, so you either have stitches, but not if you have a big chunk of tissue missing, because that you have a hole and you just have to use the pharmacology basically. But what you need to do, you need to stop the bleeding, you need to protect the wound from trauma and decrease the risk of infection, which is very high, and create the most favorable environment for wound healing, which mostly deals with reaching the correct moisture level, because if the wound is too moist with all the fluids drainage, you have like putrefaction and infection. But then if it's too dry, and then the skin, it's really hard for the skin to close, basically. So Galen talks about the same, uses basically the same parameters. And this is in his book on the therapeutic method. And so he classifies the wounds according to the origin. So you can have a trauma, but you can also have an internal origin. So you can have bad fluids, like an excess of blood that becomes corrupted, like in the case of varicose vein. And then you have different levels of tissues involved, and you can have also the time, if it's acute or chronic, old or new. And then you have different healing process according to the type of wounds that you have. And somehow you have to adjust the treatment depending on the type of wound. When it comes to pharmacological treatment, Galen's pharmacology, the ancient pharmacology, is based on the idea that, yes, you have, if you have an imbalance in the mixture of the bodily part, you have a primary pharmacological capacity, which means that the drug is either warming, cooling, drying, or moistening. But then you have secondary capacities. So you have something which is like purging or pain killing, sweat inducing, cicatrizing. So you have all different properties that can affect many different processes or the bodily fluids or the anomaly that you have in the bodily fluids. So when it comes to wounds, the drug needs to be, first of all, drying. When Galen says drying, it means that it needs to, it's not just because in Galen's system, skin is dry while flesh is wet, which is kind of a very intuitive. But when he talks about drying, it means that you need to remove the fluid, in the wound. And then you need something which is needs to join the margin together, it needs to be cicatrized, it needs to regenerate the flesh, it needs to stop the bleeding. Then you move to the actual pharmacology, where Galen, in addition to all the theory, he has all this formulation which he collects from previous medical authority. So most of them, they are not by him. He says, I'm providing you with the most valuable, the most effective, the one that have been tested and that have been used like for centuries before him. But also you need to learn how to compound a remedy. So if you have in the middle of nowhere, you don't have a recipe book, you need to know what to do and what to use and how to combine it. When it comes to the recipes themselves, so what I did, I analyzed 160 recipes to find a pattern of ingredients. So you have this most frequent ingredient, and you combine this with what Galen explains, because he says, actually, he gives you the pattern himself. So he says you need to, the basic ingredients are, this is a simplification, obviously. This is what the ingredients are, and this is what they do in the formulations. Okay, so the most frequent ingredients are, the key ingredients are metals, mainly copper compounds, and then you have plant resins, and then you have wax and other bee products, and then you have vinegar and wine. Plant extracts are actually not very important in the wound healing plasters. And then he explains what they actually do in the formulation. So the metals, it's very clear in Galen's rationalization that metals are the true active ingredient, because that's the drying, that's the main drying ingredient, which is the principal, that's the fundamental capacities that is required for the wound to heal. And then you have wax and resins, but they are excipients in Galen's explanation. So wax and resins are added for consistency and for stickiness. There's a huge issue about what Galen means by resins, because you have some liquid, what he calls liquid resins, which are actually the excipient. And we need to provide the stickiness, because it's something that needs, it's basically like a medicated dressing, so it needs to stay in place. But then you have other resins like frankincense and myrrh, which have different properties. So they're added because they actually regenerate the tissue. And then you have the propolis, for example, which is not very frequent, but Galen uses that as an example to explain what an active ingredient is. He says the propolis is added because it belongs to the very scope of the cure. It's not added for consistency. And then I wanted to, the last few minutes, I wanted to explain how, in analyzing the science behind this, using an experimental approach is actually quite important. And it's not just because it kind of helps validating the efficacy of the substances, but it also because it helps you understand the text. So the key ingredients like frankincense and myrrh, they're plant resins. And we know nowadays, we have evidence, although the quality of the studies sometimes it's kind of very it's very heterogeneous, I would say, but they have a lot of, they've been used in traditional medicine for a long time, and they have a lot of different pharmacological activities, including antimicrobial, both the frankincense and the myrrh. And the interesting thing is that they seem to have a synergy. Okay, so when you put them together, their power is actually greater than the sum of the single powers. So they create new compounds, and that makes the combination more effective. And that's very interesting, because in antiquity, they were often used together. And then we have copper and vinegar, and they are very well-known antimicrobials. And then we have the propolis again, which is it's basically wax and plant resins. And that's, again, a well-known antimicrobial nowadays. So what I did, first was to validate these key ingredients, kind of these 5 key ingredients for which there was already scientific evidence. So I tested the propolis, I tested the frankincense, the myrrh, the copper acetate, which is the verdigris, so it's the most frequent copper compound that we have in the formulations, and then the vinegar. And they all have, this is kind of a very preliminary study because I've been in the lab just a couple of months, but they all seem to have antimicrobial activities, which are, of course, much weaker compared to a normal antibiotic. But still, they are quite interesting, and you don't have the problem of also the bacteria developing. You have less issue with the bacteria developing actually antimicrobial resistance. And then in parallel, I replicated the plaster to see what happens when you handle, all these substances. So in addition to the copper compound, the vinegar, and the myrrh, and the frankincense, I added the wax and the gum and what Galen defines as liquid resins. And I follow Galen's procedure, like the general guidelines. So I melted in a water bath the wax, the propolis, and the liquid resins. And then I pounded the frankincense and the myrrh with the vinegar. I soaked the copper compound in vinegar, and I mixed them all together. And you obtain what Galen defines as a green plaster. It's green because from the verdigris. And it's very malleable. It's there when you warm it up, and it sticks to the skin, and it's easily removable. And you really get a sense of what Galen has a lot of digression about, the ratio between the resin and the wax and the stickiness. Yes, but you need to be, it needs to be easily removable. If that's too hard, if you put too much resin. And you really, and then when you do this and you really understand what he says, it really makes a lot of sense. And these substances are also, they are horrible. They are horrible to handle. Sometimes you say, okay, maybe there's a reason why they don't use them anymore, because they stick, everywhere, and they are very hard to remove. But you really learn a lot by handling the substances. And then I tried also to see if the plaster itself that I obtained has antimicrobial activities. And so I used a kind of an assay that is used mainly for semi-solid substances. So basically you have this gel, and you dig a well in the gel, and then you put the substance in the well, and then you plate the bacteria on the top, which is the yellow part, and then whatever you have in your substance diffuses through the gel, and if it depending on the chemistry, obviously, and then it can kill the bacteria around. So where you see the blue clear area, that's basically where the growth of the bacteria is inhibited. So there seems to be something which diffuses through the gel from the plaster and actually kills the bacteria. So this was just the first attempt, but it was to have an idea if actually you have something going on when you combine all those together. I just want to spend a couple of minutes to. I have time? Okay. So among the resins that we have, we also have another resin of substances in Galen's formulation, which is pitch. But Galen has different types of pitch. So you have a liquid pitch, a dry pitch, and the pitch, what he defines as the pitch oil. Now, pitch is actually, it's a resinous substance because it's obtained from pine wood in a certain way, which has been, it's been described by Theophrastus and other ancient scientists, or not only scientists. But here you have to put together somehow different sources to understand what these different types of pitches are. And basically, the liquid pitch is what you obtain directly from the pine wood. And then but this pitch has different fractions. And these fractions apparently can be separated by a procedure that looks very much like an early form of distillation, which is supposedly distillation is a much later process, like that starts in the medieval Arabic pharmacology with the invention of the alembic. But when we came across this text, actually thought, oh, maybe we can actually try if this works, and this can actually be some form of distillation. And so we actually produced the pitch ourselves. This is a kind of a side project that I I have in collaboration with Sean Coughlin in Prague, who works on ancient perfume making, and he works with a chemist and he works on resin. And so we have this kind of interest in common. And so what we did, we produced the pitch. I'm not going into the details of this, but we produced the pitch ourselves using fat wood, so this pine, very resinous wood. And okay, I'll just skip this. So we went into the forest basically, and we used a procedure which is described. There are slightly different procedure, but the concept is the same. So basically you have this wood, which is very resinous, and you burn it somehow into a closed barrel. So it's a closed environment with a low oxygen level. So the resin in the wood evaporates and condenses, and then you find a way to collect it. Basically, that's what pitch is. It's kind of a burnt version of the resin. So we did produce the pitch, and we collect a lot of pitch. And then we tried to boil it and see if what evaporates and condenses in this piece of wood that we put on the top. Okay, so this is the procedure basically. You have the boiling pitch, you put a woolen cloth on the top, and then the rising steam is absorbed by the woolen cloth, and then you squeeze it out. That's what the pitch oil is apparently from ancient text. And so we did that kind of in a very similar way. So with the copper pot, and then we put a piece of wool. And then we actually saw the evaporation and the condensation and the absorption, but then we weren't able to squeeze much out because we kind of overburned the woolen cloth. So that didn't really work out. So we have to try that again. But in parallel, we just as a proof of concept, we wanted to see if something evaporates. Instead of using the pitch, we use the resin. So we used the resin and we warmed it up, we boiled it, and we put a piece of carded wool in the top of the flask with the resin in the flask. And we actually checked if something evaporates and condenses in the piece of wool, and then we squeezed that out and we analyzed the compound that was actually absorbed by the wool. And I finished. Okay, so this is what we did, just basically as a proof of concept. So we put the resin in the flask. This is me and Sean Coughlin in Prague and this chemist. So the resin boils and something evaporates. And then it condenses in the wool on the top. And then we squeeze that out. Yes. And we obtain something that looks like turpentine. The turpentine is the distillation product of the pine resin. So from, just from the first attempt, we actually, from the smell basically, and the and visually, we thought that this actually might look like turpentine, and now we are analyzing this to check if this is actually something that can be the distillate of the pine resin. So I just want to conclude by saying that somehow you need to acquire an historical perspective, and you need to keep going back and forth from modern to ancient. That it's there are many assumptions that are probably wrong, and sometimes you just need to say, well, maybe what we see is what Galen saw. It's not always a social construct, because a broken bone is a broken bone, and a wound is a wound, and the fluid in the wound is the fluid in the wound. And we can learn a lot. We need to somehow analyze it obviously in Galen's medical system, which sometimes doesn't make any sense to us. But then at the same time, you can analyze, you can learn a lot from the way that Galen explains the natural phenomena, even separately from the potential efficacy of what he used, which can actually still be used and can be medically applied. And the experimental approach is really helpful, not just because you kind of unfruitful way to verify the efficacy, but also because you can understand the text better. Thank you. I'm sorry.